title is very much on the essence, which is international collaboration that describes HEAP itself, but also because it's very multidisciplinary work, what we have been doing. And as you heard in the morning from Alison, very nice presentation on the whole concept and the idea, what is exposome? So in short, all accumulating exposures in the life course. And as we think in from prenatal to elderly life. And our part in HEAP and in the exposome work, we thought addressing question like what exposome can tell about, for instance, preterm birth. It is a topic which has been researched for a long time, but it's still unknown the clear factors causing it, but we know that environmental factors play likely a very important role. So integrating that in the HEAP exposome work came quite naturally from the University of Oulu, because we have a long tradition on the cohort work and maternity cohort work, looking on population health from pregnancies till older age. One of them is the famous Finnish Maternity Cohort, which is an old serum bank of about 1 million women who were pregnant in Finland. And so this is the baseline, and therefore we designed the proof of concept design, which you heard a little bit on in the morning from Alison's talk as well, to follow up 100 pregnant and non-pregnant women for at least 4 months. And again, I emphasize that this is a proof of concept, so we wanted to learn how we can do it, what is feasible, and how we can do it better. Of course, you cannot do it alone, so the recruitment and ethical evaluation were done in Oulu, but obviously in a close collaboration with Karolinska and Stanford University, as you heard. What we did was we wanted to measure the personal exposure among these pregnant women. Ideal use was this wearable device, or exposometer, we call it, where we have the cartridges. The device can be controlled, the user with a mobile phone. And as the picture shows, I have a pointer here. It's very easy to wear. In the beginning, we heard that there was questions how this could be built differently or better, but after the 4 months follow-up, actually most of the women were pleased, said that it was not that difficult, and that then they were asking, so I'm very interested to know what is my exposome. We did the collection of the samples for biological profiling, chemical profiling, and the device also gives you the coordinates, so we get the detailed environmental location. It sounds a small cohort, but 100 women non-stop wearing the devices and manage the data flow, the help desk work is a bit of a demand. Here's a picture of the 9 research nurses who were actually managing it. Enrollment in 11 cities in Finland, about 30 health stations, which we used. On total, we contacted a bit over 300 women, and more than 100 accepted to take part. And in general, it was received very positively. So I see a lot of potential in this type of monitoring, even for pregnant women. Again, you remember this from the earlier talk. This is the design where we have the personal aerosol sampling, but also for having reference data analysis from urine, saliva, serum, and also cardiac rate monitoring for further correlations is possible. And the current status is that we have the whole cohort recruited and finished their monitoring. We have the 25 individuals where we have pilot data, where we did the whole protocol to test our methods and work. And hopefully we have the full cohort more or less ready this summer. As heard, for the biological profiling, we have extensive sequencing automated in collaboration with Stanford. And the unique here is not only that the state-of-the-art Ultima sequencing is fast and cheap, but it also gives you rather long sequence reads, which helps you in understanding what you have in your minute low biomass sample, which I come later on the talk. The data in general looks like this. So you have the location mobility data, which you can use plotting on different environments, biological profiling in various visualization, and the cardiac rhythm at individual level. Also, we have the chemical profiling that is mostly done in Stanford, and I won't go too much in detail on that part. But at the same time, we were building the data management system in the Finnish IT Center and CSC. So using these devices for the research is one thing, but you also need to manage quite a large flow of data with its own requirements. So this is another very much collaborative work we have done during the HEAP project. Then we've been also building different pipelines. You already heard Alison's talk on their pipeline, focusing on describing the full essence of the biological content. We've also been building pipelines at CSC, and there the focus mainly is to, so here is the Stanford pipeline, which focused on de novo assembly and then describing any trace of known biological taxa. In the CSC pipeline, we also used de novo assembly, but the main purpose is to try to go deeper on that fraction that we can define the strain level variation in the exposome. Here, basically, it means that you find the depth of the sequence data you can measure either gene level or genome level. And why this is important, we think particularly because if you want to understand the pathogenicity of the infection, you want to know what is the variation for the pathogen. As an example, here's a viral family infecting humans, and only a small fraction of those are pathogenic. And the same applies on many bacteria. So, and again, looking on metagenome, the challenge is that what you actually detect and what is relevant. Here is a recent major paper on gut microbiome, where a famous Nature paper was retracted because most of their detections in the cancer genome atlas was not actually exact detections. And using that example, we also tested the CSC pipeline with the TCGA data, where you can see on the red and yellow, which are very likely false positive or background noise, and the blue one is the candidates of a good species level taxonomy. So these were the preparation on the technical part, and now I focus on the biological profiling of the pilot data. So 25 individuals, 2 months, and we looked on the DNA and RNA of that data set. from the plant exposure. On the left, you can see with the color, the red and the orange, that is the fraction that we cannot exclude that is it a true trace or is it a false positive. So there's 65 plant species taxa, which we can for sure say that that goes across the whole data. And 6 out of those, they clearly define this strong pattern of seasonal variation, which is defined on the PCA, which is blue is winter and red is summer months. And another layer looking only on the bacteria, out of about 6,000 taxa detected, there's a clear fraction which is those good candidate species taxa, and out of those we have 177 significant candidates. We can see a nice cladding between winter and summer months, which is on the PCA in the center, and then also shown on the plot on the right hand side. And the same bacterial distribution. Unfortunately, the picture does not show it well. The main result there is that you have a strong low abundance taxa distribution on the summer months on the bacteria, whereas in winter, it seems that you have very strong dominant taxa. Then when you look on the viruses, again, you see that on the yellow, you have a huge cloud of likely false positive virus taxa, whereas we have 37 very clear good species taxa. And from those 32, strongly separate the winter and summer months. And interesting here is that compared to bacteria or fungi, which are more diverse, more diverse on the summer months, viruses are on the other side. So on the winter months, viruses are more diverse in detected with the UPAS. And again, going on the fungi level, we see that about 40 species taxa are clear, trustable signals depicted as a blue dots. And when we look at the seasonal variation, the winter month shows less diverse distribution of those taxa, whereas on summer months it's a lot more diverse. And here it's also shown on the left, you should see a bar plot showing that the seasonal variation is rather strong on the fungi as well. And you can see on the cladding that there's a clear clustering on the certain fungi on the winter compared to summer on the other taxa. So we observe this previously detected seasonal variation very strong already in this pilot data. And all in all, already on testing the method, we can see that this type of monitoring of pregnant women is very much feasible. We can see very strong dynamic change of all the different taxonomic layers by the environmental time point. And when we have more data, then we can also look on the location at the city level or urban or rural level. And in that sense, it seems that it's very feasible for personalized monitoring. And we truly believe that this can be used to predict changes in individuals' health in case we can define clearly what is the true pathogenic part of that exposome. And we hope to have the full cohort this summer ready for all these analyses. So to conclude this, I want to thank all the HEAP collaborators, Matti Lehtinen, Joachim Dillner, on the masterminds, and Alison Zhang and the Stanford Lab, US, for all this collaboration. Thank you very much.